I'm just one of those people who has a lot of questions, inconvenient questions, but I'm a little bit concerned about this runaway mRNA vaccine train. Now, just in case the censors are watching, I'm talking about beyond the COVID vaccines here and drifting into the realms of where this is going. And just in case you're not aware as to what is happening, this is big business. And anything that stands in the way is likely to get swept aside. So here is the reality of what is happening in the background. So you need to be aware of it. So here we have the situation. This is from the WHO. This is a new initiative launched to advance mRNA vaccine development against avian influenza H5N1. All great stuff. They're preparing for the next pandemic. And then you have here, US will transition all to trivalent flu vaccines for 24-25, and they're looking to use mRNA technology for that. Well, I guess great news for the investors. Here we have the HSS provides 176 million to provide develop pandemic influenza mRNA based vaccine. So again, a lot of money is being driven into that. You have here the UK's first norovirus mRNA vaccine trial is being launched in the next couple of weeks. And you have here again the WHO's push for global mRNA vaccine access. They were worried here that they didn't have enough access to mRNA vaccines in the COVID pandemic, and they were concerned that they didn't want this to happen again. And in the background, they're looking against mRNA vaccines for HIV, rabies, chikungunya, and other pathogens in the pipeline. This is going full speed ahead. Don't get in the way by just asking questions. But the truth is that I do have some questions, some very important questions. I'll mention a simple one that I'm not going to focus on today, but the fact that they didn't get long-lived immune cells in the bone marrow after mRNA vaccines, to me is a pretty big red flag. That's a problem. But here is the basic premise that you have to understand when you think about vaccines. Vaccines are different from drugs. So you take a drug when you are sick. If you have hypertension or if you have an infection or you have some disease, you take a drug to try and get better. And if you don't do anything, you will remain sick. Okay? The difference with a vaccine is that vaccines are preempting disease. So they are given to people who are healthy. And the healthy people, then, if they are exposed to an infection down the line, they are then protected. That's the premise of a vaccine. Now, what you don't want to happen is that you give a vaccine to a healthy person to prevent disease in the future, but you make disease when they get the vaccine. That doesn't work. So that's a problem. Now, that's where I have concerns with regards to where we are at the moment. And I'm going to take you through a few slides that I have prepared. And it's from my advanced COVID presentation. Just in case you're interested, if you look in the link below, this was me capturing all the research I've done over the years. And this is almost 60 modules of information going through all aspects of COVID pandemic, because I realized if I didn't put it down, I would forget some of it. And I keep on adding sections to it so that this is a process in evolution. So this, if you're interested in lots of information, this is all here, and I'm continuing to expand on it. But in the process of doing that, I've put together some pretty important slides that help me to understand a little bit more about what is going on. So I'm going to explain a little bit about that to you. So let's start with some basic things about the technology and how it works. I've got here just a picture of the different types of vaccines. I've shown this previously. The principle is simple. AstraZeneca and the J&J vaccines, they use an adenovirus um, platform. So the DNA to make the spike protein is in this. It will then enter the cell where it's injected. It goes inside the DNA to make messenger RNA, normal messenger RNA, which then produces a spike protein, which then triggers an antibody response. And that's how the adenovirus vaccines worked. The mRNA vaccines, however, didn't need to go into the DNA. And so they produce here the mRNA, which is methyl pseudourinated. So it's hidden. Then it produces this mRNA, which then makes the spike protein and triggers an immune response. All well and good. The problem is, and this is where it now comes to the scientific problematic question for me, is that in order to understand how it works is that you have to understand that in every cell there are guards, and these two guards, rig1 and mda5, what they do is that any foreign DNA RNA that comes into the cell, they come and pick it up and then trigger interferon responses because they're protecting against viral infection. That's their purpose. The problem is that in order for mRNA to work, it has to get past these guards. And that fundamentally for me is a really important question as to what does that mean going forward. And so you can see here, in order for the RNA within the lipid nanoparticle to start making protein, it has to evade these two sentinels. And in the process of evading it, it seems as though it has an impact on the interferon response within the cell. And that to me is really problematic because interferon is a very, very important aspect of innate immunity. It's the warning signal. And so, one of the strange things that we have seen, and you can see it here, this was a Cleveland Clinic study where it was showing that effectively, after people had been vaccinated in terms of their booster doses, 0, 1, 2, 3, greater than 3, and this is cumulative incidence of COVID infection. Remarkably, it seems that effectively you have more probability of getting infected the more doses you have. Now, that doesn't seem flawed when you understand the impact, therefore, that it is having on the interferon production inside the cell. And so that's a big aspect of one of the problems I have is that I still don't know if we know what is happening to these sentinels even after this is supposed to be out of the system, because I kept on asking the question, nobody knew. Suppose the lipid nanoparticle, which we know circulates, say ends up in a precancerous cell. And that cell, because it doesn't die, it's already a cancerous cell that's evading the normal death signals, then what happens to the mRNA? Does it just keep on producing? Does it never stop? I don't know. And so when I see the speed at which they're developing these things, I'm thinking, you need to put on the brakes. You need to do some fundamental research. Make sure the technology is fit for purpose before you give it to healthy people, which may cause disease. But it seems that there's too much money involved. And therefore, it means that, well, your health is secondary to the financial implications of stopping this runaway train. Here is a study that was done. And this study was looking at methyl pseudouridine, friend or foe in cancer. Now, in case you don't understand, the methyl pseudouridine, so how it works is that pseudouridine is what they got the Nobel Prize for. They replaced the normal part of RNA uridine with pseudouridine. in order to evade the immune system. But they took it a step further for the COVID vaccines and they used N1 methyl pseudouridine, even more evasion because they found it was better evading the immune response. When they looked at this particular paper, and I've just highlighted here a couple of things here. So it has been discovered that the mRNA vaccines inhibit essential immunological pathways, thus impairing early interferon signaling. So this is what I was saying. This is what happens because it blocks, interferes with these pathways. Now, what they then did is that they used 100% methyl pseudouridine in a trial with some animal studies. And what they found is that in this cohort, using a melanoma model, they found that it stimulated cancer growth and metastasis and spread. Now, it's the fact that they used 100% in the study. We don't know if it would be the same if it was 75 or 50%, or 20%. But the point is that this seems to have an impact even on the way how cancer operates. And it makes perfect sense when you think about it because, as I said, it has to evade innate cellular immunity. I've got an image here that I will show you. And this is how it looks in my mind when I did it, is that for the mRNAs to be able, this is the modified RNA, normal mRNA is destroyed very quickly. So it doesn't seem to be as much of a problem with the adenovirus vaccines. But modified RNA with the methyl pseudouridine has to tie up the sentinel, the security guard, in order for the cell to produce spike protein. And as I said, when you think about it overall from an immune perspective, it just has some red flags that need to be addressed. And this is part of the reason why I said that they need to do long term studies. You have to understand that in most of medicine and science, it's one thing that if you are using a drug, say for instance, when we had Vioxx, the Vioxx issue, we were using this drug for pain. It was a COX-2 inhibitor for pain. It had less impact on the stomach, so it was great. But one of the issues that then happened is that they found that using this drug tended to cause cardiac events. So it was an unanticipated impact of the drug. It's not any different in a sense with vaccines. Even though most vaccines have their impact within a short period of time, they can also still have longer term impacts. And when we think of the swine flu vaccine in 2009, a number of children ended up with narcolepsy, which took almost a year to be diagnosed. So we do have to find ways to make sure in the long term there is safety. I just can't understand what's so difficult about that. Is that we've used COVID vaccines in for about 3 years. Why would you not ensure that you have the adequate levels of safety by just waiting a little bit longer? Look at the 5-year, the 10-year studies to try and understand, are there any unknown risks that we have to be aware of? It's inconvenient, I know, and I mean it slows down the process. And as I just in case you're joining now, just to see what we have in the pipeline, this is why I'm asking the question. This is a runaway train. Here we have the WHO pushing for global mRNA vaccine access. They're looking at a number of candidates: HIV, rabies, influenza, chikungunya, and other things in the pipeline. The UK has just done the first norovirus mRNA vaccine. Now, this may be useful because norovirus causes a lot of problems, but as I said, you don't want to use something that could cause unanticipated issues that you just didn't know about because you didn't take the time. Here we have the HSS, 176 million to develop mRNA influenza-based vaccines. The US is planning to transition all of its flu vaccines using mRNA as well. And you have here the again, this is a WHO new initiative launched to advance mRNA vaccine development. This is going on and on and on. And if you don't ask any questions, well, you can't complain in the future, as you have seen with the pandemic. Just remember, all I'm asking for here, I'm not even saying that they shouldn't use the technology, okay? I'm just saying, just take some time, reflect carefully on what you have done. Look at the positives, look at the negatives, and ensure that you are doing your utmost to minimize the risk to the population that you are apparently there to protect. Have a great evening. Remember, like, subscribe, join our YouTube membership program, and if you're interested in our courses, look in the description. Have a great evening.